So this demonstration is just to help kind of give you a warm-up and walk you through some of the problems on the Evolutionary Genetics Lab. You should have already read through the Evolutionary Genetics cheat sheet. It's got all the basics and kind of helps give you a review of some of the rules for solving genetics problems. And so this comes from the first sheet of your Evolutionary Genetics Lab, where we're working with this hypothetical group of monkeys called the leakies. And right now we're just dealing with the spotted trait. And so here we've got our trait that we're interested in, and we're using the letter S to stand for the gene for that phenotype. Now remember, we use a capital letter to denote the dominant condition of that gene, and we use a lowercase letter to denote the recessive form. So two alleles in this gene, dominant form is spotted, recessive allele is non-spotted. And what that means is there are three potential combinations that those two alleles can join into. One would be two of the dominant alleles, homozygous dominant. We could get one dominant allele with one recessive allele, which is heterozygous dominant, and then two recessive alleles, which is homozygous recessive. Now for the phenotype, remember that anytime you get at least one copy of a dominant allele, that's going to cause the dominant form of that gene to be expressed in the phenotype, in the physical characteristic. So this time we've got two dominant alleles, that's going to produce a spotted monkey. Likewise, since we have one dominant allele paired with this recessive, it means that dominant form is going to cancel out that recessive allele and show up in the spotted phenotype. And now in this genotype here, two recessive alleles, this is the only way in which you can get this recessive condition to show up in the phenotype. So when we get two recessives, that gives us a non-spotted monkey. So on your lab worksheets for A and B on question 1, you can fill out that information based on the demonstration here. Now, using this information for question number 2, this is asking you to predict the outcomes, both the genotypes and the phenotypes, for a mating between two leaky monkeys, male and female. So it tells you in the information that's provided that we've got a male that is homozygous dominant. Okay, stop right there, and on your lab worksheets, write in the two alleles that make up his genotype. Homozygous dominant. That means two of the dominant alleles. Homo means same, and in this case, the dominant part of that expression tells you that it's two of the dominant alleles. And for the female, it says that she is heterozygous. We don't even need to say the dominant at the end because we know that one dominant allele will cause that dominant form of the gene to show up in the phenotype. So heterozygous always means one of each allele, one dominant, one recessive. So right next to the female, write her genotype, one capital S for the dominant allele, one lowercase s for the recessive allele. Now, what we do is we use this device called a Punnett square. It looks like a tic-tac-toe board. And we divide the alleles from the genotypes of both individuals into one allele in each box. So we'll start with the male. We put one of his alleles in the outside box here, and the other allele from his genotype into this outer part of the box. Now for the female. One allele and the other allele from her genotype. This actually represents the production of the gametes, or the sex cells, those cells that are used in reproduction, where only half of the genetic information is going into each one of those cells. Sperm in males, eggs in females. Those are the gametes, the reproductive cells. Okay, they have to have only half the genetic information of the individuals involved in reproduction. Why? Because they're going to combine together or recombine to form the fertilized egg or the zygote. And that's where we get those terms, homozygous, heterozygous. It's telling you what combination of alleles that zygote inherited from the parents. So, in the inner boxes in the Punnett square, we're going to combine these alleles together. And we do have some rules. You always have to write the dominant allele first in genetics problems. So we're going to combine these and we get 2 dominant S alleles. In this box, we get a dominant with a recessive. In this box, again, 2 dominant alleles. And in this one, a dominant and a recessive. Now, it's going to ask you for the ratios in which these genotypes and the phenotypes that they produce appear. So first of all, we have 4 inner boxes here, but we only have 2 genotypes that are possible. We have homozygous dominant and we have heterozygous dominant. So there's only 2 genotypes possible. And you can count how many times that genotype appears in that square. So 1, 2 would be 2 out of 4 times. And for this genotype, heterozygous dominant, it's also 2 out of 4 times. Now, for the phenotype and the ratio, all we need to know is that we translate the genotypes into the phenotypes that it produces. So, this combination of genes or alleles, this genotype produces a spotted monkey, and this genotype produces a spotted monkey. So in this case, sorry for my pen, we're getting a 100% chance of the offspring being spotted. So in this case, the phenotype is 4 out of 4 or 100%. So no chance that these 2 monkeys will produce a non-spotted monkey. There's just too many dominant alleles that are joining together to cancel out those recessive alleles from the female. Now, on page 2, you're dealing with some intergenerational data. So we've got the parents and we've got the offspring, and we're kind of working backwards based on the information that is provided. So on question number 3, it's giving you information that we're working with the spotted trait. So again, dominant allele is for the spotted condition, recessive allele is for the non-spotted condition. So you'll notice that on your worksheets that it's asking you to begin with listing the phenotype. So you're probably going to have to use a magnifying glass or something to actually see those little images of the monkeys. But look really closely at whether the monkeys are spotted or non-spotted for these family members. So we'll begin with the phenotype, and you're just going to write spotted or non-spotted. So the father is spotted. Mother is non-spotted. Junior is spotted. And baby is non-spotted. Now, are there any family members in this example whose genotype we already know? Well, the answer is yes, because we know that the only way you get the recessive condition to be expressed in the phenotype is when you get 2 recessive alleles paired together. So, we have our non-spotted family members, mother and baby, who are both non-spotted. So we already know what their genotype is. They're both going to have 2 recessive alleles. So we can record that information below the phenotypes. Okay, now for father and junior. Well, we know they're both spotted, so they have to have at least 1 dominant allele. So we'll start with that. Okay. And now this is where we have to look at this intergenerational data. We know that baby got 1 of his recessive alleles from his mother, but where did the other recessive allele come from? It would have had to have come from dad. Okay? And likewise, since Junior is spotted, we know that that dominant allele came from Dad. And since recessive alleles are the only alleles that the mother can pass on, Junior has to be heterozygous dominant. So both father and Junior have heterozygous dominant genotypes and are therefore both spotted phenotypically. Okay, now in family B, you'll notice there are some differences in the parental generation and in the offspring from family A. So I went ahead and wrote the phenotypes up there. In this family, we've got a spotted mother and a spotted father, but we've got Junior, who is non-spotted, and Baby, who is non-spotted. So again, we know that both of these family members can only be homozygous recessive. But how can they be non-spotted with two recessive alleles while their parents are both spotted? Well, again, we know that the parents have to have at least one dominant allele in order to be spotted, and each of them must also have that recessive allele to pass on to the offspring. And so this brings up a really good point, which is that this process is entirely random in terms of which alleles go into those reproductive cells that get inherited by the zygote. And so if we put these same genotypes of the parents into a Punnett square, right, we'll put Dad along this side and Mom along this side. If we work this problem in the Punnett square, we'd get two dominant S's or homozygous dominant. Here we'd get dominant and recessive. Here we'd get heterozygous, dominant, recessive, and here the homozygous recessive. So although there's only a 1 in 4 chance of the offspring inheriting two of the recessive alleles, that's a really important thing about the Punnett square, is it's not predicting probabilities, it's predicting possibilities. It's just showing you what genotypes are possible from these two individuals when they produce offspring. So it's not as if they had 4 baby monkeys that 1, 2, 3 of them would come out spotted and only 1 would come out non-spotted. They could have 6 baby monkeys, and all of them could wind up having inherited those 2 recessive alleles, or perhaps they could have baby monkeys that don't have any recessives and could be dominant, all homozygous dominant or all heterozygous dominant. So it's a completely random process in terms of which 2 alleles recombine in the genotype for the zygote. Now we're going to work with the ABO blood system. And as you can see, there are 3 alleles involved in this human characteristic, which determines the type of blood that you have. So we've got A, B, and O in this 3 allele blood system. And within this, we know that A is a dominant allele, B is a dominant allele, and O is the recessive allele. Now, A and B, since they're both dominant, they are co-dominant, meaning neither one cancels the other out. However, A is dominant over O, and B is dominant over O. So both A and B are dominant to O, but co-dominant together, meaning they don't cancel each other out. They're both going to show up in the phenotype, or in this case, the blood type. So you may know that we have 4 different blood types, or 4 phenotypes from this ABO blood system, and they are type A blood, type B blood, type AB blood, and type O. So here is where we can really appreciate how A and B are co-dominant alleles and how they're both expressed in the phenotype. So the only way to get type AB blood is to get 1A combined with 1B allele. So we know that the genotype that produces type AB blood consists of the alleles A and B. Both dominant alleles, both show up in the phenotype or the blood type. Now, looking at this information about the ABO blood system, we see that O is the recessive allele here. And we know that the only way that the recessive form of a gene shows up in the phenotype, in this case, the blood type, is when you get two of those recessive alleles paired together. So that tells us the genotype that produces the type O blood type would have to be two recessive alleles or two O alleles in the genotype, the combination of alleles. Now for type A and type B, there are some more possibilities here. Okay? So let's start with type A. There are two potential genotypes that produce a type A phenotype or blood type. The first is homozygous dominant. Homozygous means same. So in this case, since we're dealing with type A blood, those two dominant alleles are going to be the A dominant alleles. The other genotype that produces type A blood is heterozygous dominant. And that means two different alleles, non-matching alleles, or a dominant and a recessive. That's what heterozygous means, different. One dominant, one recessive. So that's going to give us a dominant A with the recessive O. And of course, because we've got one dominant, it's going to show up in the phenotype, in the blood type, and cancel out that O. Now, the same information applies to type B blood type. There are two potential genotypes or combination of alleles that produce a type B blood type. Homozygous dominant, or two of the dominant B alleles, and heterozygous dominant, or a dominant B paired with a recessive O. So now you can see that in working with three alleles, we're starting to get a lot more combinations, both in terms of the genotypes that can be formed together and the phenotypes that they produce. In other words, just adding in one allele into this system here, we're getting a lot more variation in phenotypes and in genotypes. 6 possible genotypes, 4 possible phenotypes. Okay, now for the dihybrid cross. Now this time we're working with 2 monogenic traits. In this case, characteristics that Mendel himself worked with when he was working out the laws of inheritance, experimenting with garden pea plants. And so these are 2 of the characteristics that he worked with: seed texture and seed color. So for both of these conditions, the dominant form for seed texture is smooth, and the recessive allele produces a wrinkled seed pod. For color, the dominant form is yellow, and the recessive condition is green. So in the worksheets, it's asking you to determine the genotype for 2 parental plants. And the information that you're given tells you that both parental plants are heterozygous dominant for both traits. So let's start with seed texture. We're going to get heterozygous dominant for seed texture, which is a capital R, lowercase r, or dominant and recessive, paired together with heterozygous dominant for seed color, which is going to give us a dominant G and a recessive G. So this is the genotype for both parental plants. And you'll see now, because we're working with 2 traits, that we've got 4 alleles. 2 alleles for each trait. 2 alleles for seed texture, 2 alleles for seed color. So 4 in total. Now the next step is to figure out the gametes, the reproductive cells. And this requires an extra step that isn't necessary when you're dealing with just one monogenic trait. Here now, because we have more alleles that need to be split into the reproductive cells, the gametes, it means there are some more combinations in which these alleles can combine and go into those reproductive cells. So we use this method kind of like foiling that they do in math, and here's how this works. So we're going to figure out the gametes. And what you do is start with taking the first allele from the first trait and combining it with the first allele in the second trait. And that's going to give us a dominant R and a dominant G, so the two dominant alleles from both of the traits. Then you take the first allele in the first trait and combine it with the second allele in the second trait, and that will give us a dominant R and a recessive G. Okay. Now we move over to the second allele in the first trait and combine it with the first allele in that first trait. And here's where we're going to break the rule temporarily, and you'll see why as we move into the Punnett square it's important. So we're going to write that recessive lowercase r first and combine it with that dominant G from the second allele, or second genotype. Okay? And then finally, second allele from the first trait combined with the second allele from the second trait, and that's going to give us the recessive R and the recessive G. So we have 1, 2, 3, 4 different combinations that these alleles from both of these traits can get into. Remember, the gametes have to have half of the genetic information from the parents. So there has to be one allele for seed texture, one allele for seed color. So 1, 2, 3, 4, and that's the information that you're going to use to plug into the Punnett square, which has now gotten much larger. As you can see, I'm rotating this over here. And we're going to take these gametes that we just figured out here and plug them in along the outer boxes of the Punnett square. Now remember, both of the parental plants have the same genotypes. So it means these gametes are going to be the same along the side and the top of the Punnett square. And you can be filling your worksheet out with this information as I go along, okay? So here are the gametes, half the genetic information for each of the parental plants. And now in the inner squares, we're going to recombine and keep the R's together, or the alleles for seed texture, and the G's together, or the alleles for seed color. And the genotypes that are going to be produced in these inner squares are going to have 4 alleles, just like that parental genotype. 2 alleles for seed texture, 2 alleles for seed color. So we follow the rules of the solving genetics problems, which means when we get to those gametes that have the recessive allele first, we're going to revert back to that rule of writing the dominant allele first, okay? But this is helping us to keep the R's and the G's together as we recombine. All right, so here we go. In this top square, we're going to get 2 dominant R's and 2 dominant G's. Okay. Now you can either move down or across. It really makes no difference. So here, we're going to get 2 dominant R's. And remember our rule, 2 G's, but the dominant allele gets written first. Here, again, remember our rules. We have a dominant R and a recessive R. Dominant goes first, and 2 dominant G's. Here, remember our rule, a dominant and a recessive. And again, remember the rule, dominant, recessive. Okay? Move along. 2 dominant R's, and heterozygous for the second trait. Might be helpful to practice saying homozygous dominant, heterozygous, homozygous recessive. So here, homozygous dominant for seed texture, and here, homozygous recessive for seed color. Here, heterozygous dominant for seed texture, and heterozygous dominant for seed color. Here, heterozygous dominant for seed texture, and 2 small g's, homozygous recessive for seed color. Here, we've got the dominant and the recessive, so heterozygous for seed texture, and homozygous dominant, 2 big G's for seed color. Heterozygous, seed texture, heterozygous, seed color. Here and here gives us homozygous recessive for seed texture and homozygous dominant for seed color. Here and here we get homozygous recessive, seed texture, and remember the rule, big G, little g, heterozygous dominant for seed color. Okay, last squares. Heterozygous dominant, seed texture, heterozygous dominant, seed color. Hopefully you can still see this. Okay, heterozygous dominant for seed texture, and homozygous recessive for seed color. Here, homozygous recessive and heterozygous dominant. And last one, 2 small r's, homozygous recessive seed texture, and 2 small g's, homozygous recessive for seed color. The next step is to determine the number of genotypes produced by this parental cross. So, although we have 16 boxes in this Punnett square, it doesn't mean that there are 16 different genotypes here. So what we need to do is go through and count each different genotype that appears in the interior boxes of the Punnett square. And what we'll do is we'll just list them here and list the different ones as we go. So we'll just start with this upper box, and we've got homozygous dominant with homozygous dominant, okay? And we just move through the box and just write down each different genotype that we encounter. So here we've got homozygous dominant with heterozygous dominant. Move on over here, we've got heterozygous dominant seed texture, homozygous dominant seed color, so that's a different one. And then here we've got heterozygous for both. Okay, so far 4 different ones. Now we move down to the second row, and we've got 2 big R's and a heterozygous. I've already got that one, so we don't need to write it again. Here we've got homozygous dominant with homozygous recessive. We don't have that listed here, so we'll write that one down. Okay, heterozygous for both traits, we've got it. Heterozygous for the first trait and homozygous recessive for the second, we don't have that one yet, so we write that down. Okay, move to the third row. Heterozygous with homozygous, we've got it right here. Heterozygous for both, we've got it. Homozygous recessive with homozygous dominant. We don't have that one listed yet, so we're going to write that down. Homozygous recessive for seed texture, homozygous dominant for seed color. Okay, keep going. Homozygous recessive with heterozygous dominant for seed color. We don't have that one listed yet, so we'll go ahead and write that. homozygous recessive, heterozygous dominant. Okay, move down to the final row. Heterozygous for both, got it here. Heterozygous with homozygous recessive, got it right here. Homozygous recessive with heterozygous dominant, we've got it right here. And that leaves homozygous recessive for both characteristics here. So, 16 potential possibilities of genotypic combinations, but it actually only produces 1, 2, 3, 4, 5, 6, 7, 8, 9 different genotypes. So there are only 9 different genotypes that appear in this square. Now, to determine the ratio, we go along our list here and count the number of times that this genotype, or each of these genotypes rather, appear in that 16 square interior of the Punnett square. So you just count, and the way that I like to do this when you're working on your worksheets is marking them off with an X as you go. So again, work off your worksheets and follow me as you're writing these down. So homozygous for both, that's right here, mark it off, it's in the square 1 out of 16 times. Okay, homozygous dominant for the first trait and heterozygous dominant for the second, we've got 1 and 2. So we'll mark those off, 2 out of 16 times that genotype appears. Heterozygous with homozygous is 1 and 2. So we mark those off, 2 out of 16 times. And heterozygous for both traits, 1, 2, 3, 4. 4 out of 16 times. Okay? See how it gets a little bit easier to locate these as you go along? So, homozygous dominant with homozygous recessive, 1 time. 1 out of 16. Heterozygous with homozygous recessive, 1, 2 times. All right. Homozygous recessive with homozygous dominant, 1 time. Homozygous recessive with heterozygous dominant, 1, 2 times. And our last remaining square, homozygous recessive for both traits, 1 out of 16 times. Now, if you add together the number of times each of these appear, it should add up to 16. So we get 3, 5, 9, 10, 12, 13, 15, 16. So all of the squares in the Punnett square are accounted for. Now our next step is to make the translation into phenotypes. I'm going to rotate this around a bit here. Okay, the phenotypes and their ratio. And what we're going to do is, just like we did with the genotypes, every time we encounter a new phenotype, we'll write it down. And so here's where we make the translation based on the combination of alleles for each trait, seed texture and seed color. So looking at your worksheets and the information you recorded earlier, if you get a homozygous dominant for seed texture, what kind of texture does that produce? That's smooth. And homozygous dominant for seed color would give us yellow. So smooth and yellow. We'll write that phenotype down. Okay. Now, we're just going to move along, make these translations into seed texture and seed color, and write each different phenotype that we come across. So, smooth and yellow. Smooth, because we've got the one dominant smooth allele here, canceling out that recessive, and yellow, so still in the same phenotype, and smooth and yellow. So we have that phenotype listed. Here we get smooth and 2 recessive alleles produces a green colored seed. So we'll write that phenotype, smooth and green. Okay, next one, smooth and green. Got it. Move down here, we get wrinkled. We have two recessive alleles that produces a wrinkled seed texture. And for seed color, two dominant alleles, which gives us a yellow seed color. Wrinkled and yellow. All right, this seed is also wrinkled and yellow, so we have it. And that brings us to the last one, which is wrinkled and green. So 9 different genotypes, but they only produce 4 different phenotypes. And now what you're going to do for the ratios is you'll count how many times the genotype that produces this phenotype appears in the Punnett square. And you've already done that step by figuring out the ratios for the genotypes. So now you're going to add all of these genotypes and the number of times they appear in the square together that produce smooth and yellow. Okay, so that's these genotypes. And we get 1 plus 2 is 3, plus 2 is 5, plus 4 is 9. So the smooth and yellow phenotype appears in the Punnett square 9 out of 16 times. Okay? Now, these two genotypes produce the smooth and green phenotype. So that gives us 3 out of 16 times that phenotype appears. You see what I'm doing? I'm adding together the number of times those genotypes appear in the square to produce this phenotype. Okay? Wrinkled in yellow, that's these two. So again, we'll add the number of times they appear, and that will give us 3 out of 16 times that wrinkled in yellow appears in the Punnett square. And finally, wrinkled in green, 1 out of 16 times. Now, as a kind of double check on your work, your ratios should add up to 16 for the phenotypes as well. So we've got 9 and 3 is 12, plus 3 is 15, plus 1 is 16. So 16 out of 16 times.